presentation will be given by Petras Juzėnas from Oslo University. He will speak about nanoparticle sensitizers for medical applications for non-ionizing and ionizing radiation. It's a collaborative work with Andrius Klinauskas, King Sun and Wei Chen from Oslo, Clemson, and Petras Universities.
Yeah, thank you very much, Chairman, and thank you very much, organizers of this conference, which made me a great opportunity to come back to my hometown Vilnius, at least for a few days. And just like the earlier speaker noticed, good evening, ladies and gentlemen. So, as the Chairman noticed, I come from the Oslo University Hospital in Norway, and I have structured my lecture so I will present the results of my group, but I noticed that some of earlier speakers today, they have also displayed their institutions. So to correct this problem, I will say a few words about my institute. So Oslo University Hospital is the largest hospital in Norway, comprising for about 40% of the biomedical research in Norway. It includes 4 hospitals, including the Norwegian Radium Hospital, which specializes in cancer therapy, including the Institute for Cancer Research, where I am working in. And also, as a part of this, a new concept of Oslo Cancer Cluster has been laid down, which will integrate the clinics, the research, the pharmaceutical industry, and the high school. So, my work is on nanoparticles, so which are small, tiny particles, and most of the research until now has been focused on imaging applications of nanoparticles, but why not try them to treat different diseases? And since we are working in the Institute for Cancer Research, our research is focused on cancer cells. So, and actually, nanoparticles is not something you read in the literature or scientific literature or media. They already can be as examples in our actually almost daily life. There are already available some products on the market, like for example roof tiles or paintings with nanoparticles, which may clean air, pollutants, also antibacterial silver nano coating. For example, to make food storage safe in a refrigerator, or to provide medical tapes to cover wounds, and also sunscreens to protect to filter out ultraviolet radiation in the sun. And actually, why we may not notice even that nanoparticles are in the product, because manufacturers, they are not obliged to provide such information yet. And also, I'm not going to advertise tobacco industry, but actually there is even available nanotech cigarettes. So why cancer is important? Because it is actually, together with cardiovascular diseases, is one of the deadliest diseases. And also statistically, it affects 1 out of 3 people in his or her lifetime. So it's really important to conquer cancer. And why we are doing research? Because there is still no ideal drug available. Available anti-cancer drugs, they have some undesired side effects. And also radiation therapy is also not selective to cancer cells. It affects normal cells to the same extent as cancer cells. So there's a niche to do this research here. So, and then we came across the idea why not to try nanoparticles to enhance radiation effects in cancer cells. And this can be done because nanoparticles are good photocatalyzers. So either under irradiation with visible light or ionizing radiations, nanoparticles can produce an exciton pair, electron and hole pair, and hole can participate in reduction reactions, and electron can participate in oxidation reactions. And ultimately, such combination with nanoparticles and radiation can kill cancer cells. In our lab, we have screened a series of nanoparticles for their effectiveness with radiation. So it can be with classical cadmium selenide quantum dots, zinc nanoparticles, and non-fluorescent gold or silver nanoparticles. And this is just below transmission electron micrographs showing that they are really nanoparticles of about 10 nanometers of size. So, and how should we start? So we started with so-called classical quantum selenide quantum, and we to some extent failed because under light exposure we hardly killed any cells. There was some tendency, but there was no significant differences. And this could be because quantum dots, as already mentioned by some earlier speakers, they enter by undersynthosis and they are encapsulated in lysosomes. And here you can see that during radiations, the red fluorescence from quantum dots goes up, showing that they were encapsulated in these vesicles. And when we do a model system, we can see that the quantum dots in solution, they generate free radicals, while if they are encapsulated in liposomes, their free radical generating capability is inhibited. So we tried another type of nanoparticles. So in this example, I present two cell lines, two human prostate adenocarcinoma cells, and silver nanoparticles. They act as photosensitizers. So actually the red curve is the cells pre-incubated with the nanoparticles and irradiated with blue light. And the black one is a control. In this example, we are investigating a new class of spherical carbon nanoparticles, so-called carbon dots. And they actually also show very good anti-cancer activity under irradiation. So they are not toxic without external radiation. And this is another new class of nanoparticles. These are similar spherical carbon dots, but with a silver layer coating, which enhances their photocatalytic activity. So we kill even more cancer cells. And this is just to compare as a normal cell line, fibroblast, and the killing effect was significantly lower. So it could be some even kind of selectivity towards cancer. This is, we also investigate if ionizing radiation, because ionizing radiation is mostly used in cancer therapy worldwide. And even these carbon dots with silver coating, they show very good enhancing ability of ionizing radiation. So cells which were pre-incubated with carbon dots and then exposed to ionizing radiation X-rays, they were more effectively killed compared to the control group. And this could be because the nanoparticles, they can generate more free radicals. Because ionizing radiation, ionizing mostly water molecules, and the ejected electrons initiate free radical reactions which usually target the DNA. So by introducing nanoparticle into a cancer cell, you actually generate more electrons and more free radicals. This is another class of nanoparticles, zinc nanoparticles. They also show some effect with ionizing radiation. And what is interesting with zinc nanoparticles, because they are scintillating under ionizing radiation. So they produce light when triggered by ionizing radiation. So maybe this could be used also for imaging purposes together with radiation. And in some literature, there are published works that gold nanoparticles are very effective enhancers of ionizing radiation, but actually this we couldn't reproduce in our lab. We don't go now into the details why this could have happened, but in this example, this figure is coming from another publication by Zhang et al. They show that the gold nanoparticles do kill more cancer cells under X-ray radiation compared to the normal control group. However, then we have, these assays are actually a single endpoint assays. We treat the cells, we make lysates, and we study the reactions. And then we have tried a so intelligent way to do it. It's a system based on specially designed culture plates, where the cells can grow for practically a few days, and then we monitor proliferation, so-called live cell monitoring. And so, for example, in this case, this is a The black curve is the control cells growing here for 3 days, and the green or the red curves are irradiated with X-rays. And in this example, actually, we did not kill more cells with pre-incubating them with gold nanoparticles. And the reason could be quite simple, because these specially designed plates, they have golden electrons. And gold is a high atomic number material, and of course it absorbs efficiently radiation. So we have modified the protocol, and then we first irradiated cells, and then incubated them in this impedance culture plate. And then we got the significant reduction in cell proliferation, the red curve. So to make this long story short, the conclusions are quite simple. Actually, the nanoparticles can act as photosensitizers, so under ultraviolet, visible or near-infrared radiation, or they can be also radiosensitizers to enhance effects of X-ray or gamma rays. So I would like to acknowledge my small group, which is pretty international, with Lithuanians, Norwegians, and Russian in the group, and also international collaboration partners from the United States and Portugal, Belarus, and also I would like to say special thanks to my mentor, Professor Richard Svetonskas from Vilnius University Oncology Institute, who is actually present here in the audience. And I have to also acknowledge financing institution, because it's very important also in science, the Norwegian Research Council, the Research Foundation of our institution, the Regional Health Authority, the Norwegian Cancer Society, and also the European Erasmus Student Exchange Program, and some smaller foundations. And thank you very much for your attention.
Plenty time for questions. What size of nanoparticles of titanium which are in sunscreens?
Actually, I have not been investigating the qualities and properties of the sunscreen, and it is not specified on the packages. But what I heard from other colleagues who are working, it could be from a few nanometers to 100 nanometers.
Yeah. Yeah.
Can you do selective killing by using particles that will go in one type of cell and not in another type of cell?
Actually, both probably yes and no. On one hand, if we attach targeting ligands into nanoparticles, so of course we can target particular cells, normal or different cancer cells, because they express different surface proteins. Of course we can target. But on the other hand, if we attach antibodies, proteins, other ligands on the surface of the nanoparticles, we can change their chemical, their physical properties, so they may not then generate free radicals. So it's a bit tricky situation.
One more last question. If it is not the case, let's thank the speaker and all the speakers of the session. Next session is dinner and conference will resume.